go ahead and get started. My great pleasure to introduce the guy who you can't see up here, Alec Hirsch, expert in Flaviviruses from the great West Campus of OHSU. Those of you who don't know about that, that's where the Vaccine and Gene Therapy Institute is and the Oregon National Primate Research Center. And they do, in my completely biased opinion, really nice work out there, and they know way more about Flaviviruses than I do. So with that, Alec.
Oh. Okay, right. So the topic today are the Flaviviruses. So the vital statistics of the viruses, these are positive sense RNA viruses, single piece RNA genome of about 10 to 11 kilobases. They're enveloped. They are all classified as arboviruses, that is arthropod-borne viruses, so they're transmitted by mosquitoes or ticks. Some of the more famous members of the Flavivirus family include yellow fever virus, West Nile, dengue, Japanese encephalitis, or Zika virus. There are also some kind of nasty tick-borne encephalitis viruses within the Flavi family that are out there lurking in the woods. Then there are other more distantly related members of the family Flaviviridae that fall under categories of different genera. So the Hepativiruses, which is entirely the Hepatitis C viruses. And there's also a class of viruses within the genus Pestivirus. These are largely viruses of animals, but some of which actually have a fair amount of animal health and agricultural importance. Next. And I can't remember which is the next slide. That's it. Okay, so the basic outline of the lecture, we'll sort of start with the virus itself, the structure, the mechanism of the life cycle going through binding and entry, replication, translation, and making of progeny viruses and release. And then at the end, sort of more of the history and pathogenesis of select number of Flaviviruses. I used to always do this with the discovery and disease first, and then I would never have time to get to the other stuff, so hopefully this order works. Basic structure of the Flaviviruses. So what we're looking at here is actually a depiction of the viral envelope. So although it's an envelope virus, where you would normally think of the viral membrane as kind of a lipid bilayer with studded with multiple proteins. This, although the lipid is sort of underneath it, the surface is actually coated with a very regular arrangement of individual proteins. So these are all the E or envelope protein, and shown here, each one of these is an envelope dimer. So there's a single, we'll look at this in a little more detail later, but that's a single envelope protein there and a single one sort of lying next to it here. And these form this regular structure so that the intact virion has 180 monomers of the E protein, all of them coating the surface of the virion. So you get this sort of icosahedral symmetry, which gives you can define as a T equal 3. You don't have a 3D printed Flavivirus yet, do you? Okay. But maybe someday he'll get one. And you should get mature and immature forms too. But anyway, so this is what it looks like from the outside. But remember, when you're dealing with this icosahedral symmetry, it's not the capsid surrounding by the envelope. This is the envelope. Well, we don't know too much about what's inside. I mean, we know what's there. It's the nucleocapsid protein, or C, and the RNA genome, but the structure itself is fairly undefined, and it may really just be sort of more like the genome's kind of wrapped around capsid structures, but it's much less regular than the outside of the virus. So this is in more detail look at the envelope protein. So again, this here is one envelope protein, here is another, so we have this dimer. The thing is normally divided into, or considered divided into 3 domains. Let's see, that would be 1, 2, and 3 here. Domain 3 is responsible largely for binding to the receptor. The receptor for most of the flaviviruses specifically is not really, have not been identified. There's early binding to sort of charged, non-specific binding to charged proteins on the cell surface, followed by a receptor entry. So just before I get ahead of myself, let me just sort of quickly just give you an overview of the whole life cycle. So as we're talking about now, we have binding and entry into the host cell. The virus actually is taken in whole by receptor-mediated endocytosis here, where it winds up in a low pH endosome, and the pH triggers a conformational change in the envelope protein, which I'll go into in just a little bit, releasing the nucleic acid into the cytoplasm. Okay. So as I said, that's a positive-stranded RNA virus, so that is translated into a single protein, which is then cleaved up into functional subunits by viral and host proteases, and again, we'll go into that in a little more detail. And those proteins include proteins that are required for replicating the viral RNA, so you make sort of complementary or negative strands to the genomic RNA. Then you make more positive strands of the genomic RNA, and these positive strands are packaged with the capsid back into the ER, and then these viruses are released. And there's a further protein in the membrane called PRM, which when it's there, the virus has this kind of studded irregular appearance. And then this is cleaved by the host protein furin, so the M protein, which is normally, PRM is about 25 kilodaltons, and then the major part of that is removed right before egress, leaving just a small bit of M, which is about 8 kilodaltons remaining in the virion. And then after this cleavage, then the envelope proteins form that regular dimer-dimer structure that I showed you at the beginning. So entry, again, coming back to the first part of the viral life cycle. As I said, you have low pH mediated or so receptor mediated endocytosis over here, and then you get to the endosomal vesicle. The pH doesn't need to be that low. It's just around 5.5 to 6 triggers the conformational change, which allows fusion with the envelope of this of the endosomal vesicle and release of the capsid and viral nucleic acid. Cool. Next, right. So the point is that we need to get into the cell. You still wind up in the endosome. So it's the fusion on the endosome, not so much the plasma membrane, to get the nucleic acid into the cytoplasm. So a little more detail on the envelope protein. Shown here again, domain, the envelope protein in its dimer form upon the low pH fusion, you get sort of this, the next slide I think is better for this, right? So you have the 3 domains here, 1, 2, and 3 upon the low pH environment of the endosome. You release this little tip of domain 2, which is the fusion peptide that inserts into the target membrane. Then that conformational change results in a further sort of reorganization. So the envelope proteins have these transmembrane domains in the viral membrane. So as this reforms, the fusion peptide and on this end and the viral transmembrane domains tend to pull the membranes into opposition of each other. The dimers then sort of reorganize to form these trimers, shown here. So these are now trimers, and then eventually you pull the membranes together and form this pore by which the capsid nucleic acid can then enter into the cytoplasm. Don't worry about all the numbers here, but a lot of this and various intermediates were figured out by specific monoclonal antibodies that bind to different domains. That's what all these are showing, antibody binding sites. So, you know, I could go on for an hour about just the work done to sort of figure this process out. But a lot of it was done using tools like, you know, blocking just individual domains and seeing at what point is the virus hung up. Okay, but so once we're in the cell, then the virus has to go about the business of replicating itself. So you put the genome in the cell. As I said, this is a 10 to 12, 10 to 11 kilobase single-stranded RNA. It has a cap, but no poly-A tail. Some of them, not the genus Flavivirus, like Dengue, West Nile, Zika virus, but the Pesti and Hepatitis C viruses actually have, instead of the cap, they have an iris or internal ribosome entry site at the 5' end that mediates translation. Similar, I don't know if you guys have done any picornavirus stuff, but you know, similar to that. There is a 3', actually a fair amount of structure in both the 5' and the 3' ends of this genome, which is still not, I think, completely defined, but there a lot of these structures are required for proper replication of the viral RNA. Let me just go back. Then further, as I said, you translate a single viral polyprotein, which is cleaved by different enzymes into 10 functional subunits. So some of those enzymes are cellular. So the ER resident signal peptidase is cleaves largely the structural proteins of the genome, the capsid from the membrane from the envelope protein, and then the virus itself encodes a protease which is made up of the viral NS, that's non-structural 3 and 2B proteins that largely cleaves the rest of the genome into the functional subunits. So this is a closer look at the genome itself. Not to scale, because most of it this is about, you know, 9 to probably about 80 to 90% of the sequence is this region here. But if you look closely at the 5' and 3' UTR, you can see all these conserved structures, which are found at least in most of the, you know, the genome Flavivirus viruses. And there's been a lot of mutational analysis of these things that show that you know, various viruses, once you just mess with any one of these stem loops, tend to either be further attenuated or totally unable to replicate. Some of the sequence elements that are of note are there's actually a cyclization sequence, so complementary sequences here in the 3' UTR, and also which is complementary to a sequence here in the 5' UTR, shown right here, implying that the virus actually circularizes when it comes in, and that's required for its replication. And a lot of detailed genetic work has shown that if you mutate one of these cyclization sequences, you stop virus replication, but then if you change the other one so that now you can get, repair Watson-Crick base pairing, then you can restore it. So it's actually that complementarity, not necessarily the sequence itself, which is responsible for virus replication. And then you have all these stem loop structures with Watson-Crick base pairing over here. And these are actually somewhat dynamic structures, so they're not always in this shape, but can, may actually sort of change depending on the environment or proteins bound to this part of the RNA. So as I said, a lot of this work, this stuff makes my head hurt, but there are a lot of people who have done quite, careful analysis of all the sequences in both the 5' and the 3' UTR and how they interact with each other, and it's kind of cool. But then if we zoom in on the coding region, which is the center of this thing here, I keep hitting the wrong button, sorry. So zoom in here. So again, this is the organization of the genome, 5' to 3', into those 10 functional protein subunits that I mentioned. Yeah. Are conserved in. Right. So as far as I know, all of the mosquito-borne Flaviviruses, so Dengue viruses, West Nile, all have this structure or these structures or something very close to it. That's correct, but I don't know the answer to that question. I don't think, for the most part, you would say that they're is particularly rich in the other one. There are probably a few little areas, within this structure, again, wrong button, where you do see a particular AT or GC, enrichment, but overall, I would say that the structures are not biased to one particular bonding pattern. Yes, thank you. AU. Sorry. Anyway, so the proteins encoded by the genome are here. Okay, so the 5' end is the structural region. That's the capsid protein that associates with the nucleic acid within the virion. The PRM protein, which I mentioned before, which functions largely as within the cell before maturation or that furin cleavage event, is something of a chaperone for the E protein, which is the envelope that we discussed. Then there are all these non-structural proteins. Let's go to the next one. Yes, so NS1, all of these are more or less essential for virus replication. I mean, there are only 10 of them, so you can't really carry a lot of, dead weight. NS1 is secreted, and actually in Flavivirus-infected patients, you can find during viremia also a large amount of the NS1 protein circulating within the serum. The actual role of that secreted NS1, I think, is largely unknown, but in some cases, like in dengue virus, it does seem to have a role in actual pathogenesis in creating some of the manifestations of dengue hemorrhagic fever. So 2B and NS3 are the viral protease. NS5 is extremely important and is a methyltransferase and RNA-dependent RNA polymerase. So this is the enzyme responsible for copying the viral nucleic acid into the negative strand and then back into the positive strand RNA genome. Shown here in the black stars are the host cell protease cleavage sites. So this is, the virus is translated along the membrane of the endoplasmic reticulum, so that some of the proteins are on the ER lumen side and some on the outside. So the ER lumen side are going to be the ones that sort of see the outside of the cell, so either the viral envelope or that secreted protein NS1. So, and then these are separated from each other by proteolytic events in the ER by the host protease. And then this NS2B3 protease first autocleaves itself, and then it's active and is able to cleave these different spots here shown by the green stars, and also releases the capsid from its transmembrane domain. Remember, you're moving in and out of the ER, so you often have to span the membrane. So these are either fairly hydrophobic, membrane associated, or they have domains which have to be removed if they're going to be fully soluble. So just back to the life cycle to remind us to where we are. So I guess we've gotten, which is the one I want, into the cell. We've replicated or translated the protein in around the ER. Then the NS5 polymerase makes the nucleic acid strand. So then you have to assemble the viruses. So this is again done in very close association with the with the cellular membranes. Okay, so the ER, although there's largely it's the endoplasmic reticulum, but there is a lot of crazy rearrangement of cellular membranes during flavivirus infections. So sometimes it's a bit hard to look at a flavivirus infected cell and know exactly what normal cellular structure is what. But but it's been observed. This is largely the work of a lab in Germany from a guy named Ralph Bartenschlager, who's done a lot of this like really high-end electron microscopy, observing all these different structures. So these big sort of circular things within the ER, or these vesicles, which are, and then the arrowheads, I don't know if you can see it, are actually pointing to individual viruses, but using just a lot of the electron microscopy technology, and then computer modeling to take all these slides of consecutive pieces through the cell. That lab has done this 3D modeling to get down to this idea of so that the ER sort of expands and looks like this. So you have this little sort of pea pod looking structure. The viral capsid associates with the positive strand genome and actually buds into the ER. So the envelope is again, as I mentioned, on the ER lumen side, on the inside. So as the capsid buds into it, it obtains the envelope on the outside. The current model is that the replication is actually sort of occurring within these circular structures, which are then topologically related to the cytosol, and then the budding goes through the ER membrane, and then you have the ER, the envelope virus within the ER. And they tend to cluster in sort of vesicles that are coated, that consist of multiple virions within them. And then this moves through the cell where it then fuses with that. Well, it moves through the secretory apparatus because it has to get to the trans-Golgi membrane where it encounters furin for that maturation process. And then finally, the vesicles reach the end where the larger vesicle fuses with the host plasma membrane, releasing the viruses. Does that generally work for most people? Yeah. So it's sort of probably like, exactly if it's like de-envelopment and then re-envelopment as it fuses and then moves out. I'm not sure, and I'm not if that's been demonstrated. Although now I want to see if I can look this up. But also because of the membrane rearrangement, there is a certain amount of movement of enzymes throughout, or sort of collapse of certain cellular structures. But generally, I think you're moving through secretory vesicles. So the envelope viruses are within another vesicle, which then fuses to the Golgi, or whatever state the Golgi's in a Flavivirus infected cell. And then another vesicle is released from the Golgi to the trans-Golgi network and so on. So that's, I believe, correct. Yeah. Yes, the secretory mechanisms are cellular, so the cell is often capable of making things and secreting different things. So that's the machinery. That's it's not being significantly altered. It is, depending on how you want to phrase it, it is being sort of co-opted by the virus so that it'll work in that, work through that to replicate itself. And then, so I have two slides that I want to just say that while all this is going on, the cell is not merely a helpless bystander. So in response to viral infection, and I don't know, do you cover interferons and antiviral replication? All right, so, but the cell does, obviously, the host has an immune response to the virus. Even before we get to the adaptive immune response, which is your B cell antibody or T cell mediated immunity, which develops sort of while this is going on, there is an immediate response, broadly defined as innate immunity, that the virus, that the cell will use to attempt to counteract the virus. And these are generally mediated by proteins, secreted proteins called interferons, which bind to cells through interferon-specific receptors, and the receptor binding induces the production of large numbers of proteins which sort of act antivirally within the cell, that act to prevent viral infection of neighboring cells, also signal to the adaptive immune system that there's a current danger going on there, and it helps prime the later antibody and T cell responses. And all that starts sort of like immediately after viral infection. So the cell is doing that, and then the virus, if it's going to be successful, needs to be able to counteract those things. So this is just sort of a slight snapshot of some of that, not all of it, involving some of the antiviral pathways within the cell. So there's shown here, if I can find Rig-I, right. So one of the major cellular proteins in this response is called, it's generally I think called Rig-I, although some people think it should be Rig-1. I'm not sure. I generally call it Rig-I. But the function of this protein is to recognize double-stranded RNA. So you have, when you have this RNA virus, single-stranded, that replicates by making a copy of the RNA, then you create this double-stranded RNA, which is something the cell does not usually see very much of. So the presence of double-stranded RNA activates this protein Rig-I, which then signals through various molecules, including this thing which is called MAVs or IPS1, which is located on the edge of the mitochondria. And then that triggers a whole, a further cascade of things that eventually winds up with a transcriptional response and production of not only these interferons, but also other antiviral proteins. You can get the same pathway going by through a protein called TRIF. Is that actually on here? That might not be on here. But that's triggered by the presence of certain pathogen-related patterns that are found in endosomes. And then there's also a ER-resident molecule called STING that also kind of feeds into the same pathway. So basically you have all these things which are surveying the cell for patterns associated with viral infection. They're ones for bacterial infection and all sorts of things as well. And then activation of those triggers this antiviral response. So the virus also has to deal with this while it's also accomplishing its replication. Some of what we know is that different viral proteins can negatively impact these things. For example, it's been shown that the dengue protease not only cleaving the viral subunits, but also is capable of cleaving this molecule STING, as well as other parts of the signaling apparatus. So, what's shown up here is what's been demonstrated, and sometimes these functions are conserved amongst the viruses, and sometimes it's fairly unique, even, among different flaviviruses have different ways of counteracting these things. And then I had mentioned that one of the responses, or sort of a big part of the response of triggering the innate immune sensor proteins, or they're generally called PRRs, or pattern recognition receptors, is the production of interferons, which is a secreted cytokine that then feeds back on the infected cell and also signals to neighboring cells as well. And so the interferon receptor shown up here, signaling through this, results in phosphorylation of these, and activation of a set of cellular kinases, which again leads to activation of transcription factors, which gives you more antiviral protein production by the cell. So it's been shown that, again, various viral proteins are capable of blocking these, the NS5 proteins, so that's the polymerase and methyltransferase, but beyond that they also have an effect on blocking these, the STAT2 protein that's part of the interferon signaling pathway. This has been shown for other proteins in dengue and different spots along the pathway, and there's probably quite a few more that have not been defined or that act in only specific cells. So the interplay between the host cell and the virus is complex. It's not that the virus just gets in, uses the cellular machinery, and makes more of itself. There's quite a lot going on during the viral infection. And this is true of all viruses, and they all have some strategy for dealing with this kind of thing. So, all right, so we've replicated the viruses. Just a few words on some of the other members of the family Flaviviridae. So hepatitis C virus, originally defined as hepatitis, liver inflammation, and when it was first discovered, there were hepatitis A and B had been identified, and so it was found to not be any of those. So it was originally non-A, non-B hepatitis, and the hepatitis viruses, or hepatitis A, hepatitis B, C, that the word comes from, they wind up in the liver and they cause inflammation pathogenesis. They have no relation to each other at all. One example that I heard once was there are sort of about as related as a banana and a toaster, in that, those are found in the kitchen, these are found in the liver, but that's about it. So anyway, hepatitis C virus, which was recognized as this chronic hepatitis virus, which in many cases would lead to liver failure or liver cancer. Well, at least generally started out in a mild acute setting, but sort of if the virus were not cleared and you get this chronic infection, that's when you have all these sorts of problems. It was, determined to be viral. There was no really good animal model. The only one that ever was used were chimpanzees, and chimp research has largely been ended in these cases. Although some of the chimps that were used in these studies are still alive and in retirement now. So in 1989, this virus, I guess, became the first one to be actually cloned or identified by purely molecular means, where cDNA libraries from infected individuals were used to identify viral proteins using immune serum as a probe, and that allowed sort of, once you had a piece of DNA, you could sort of use that as an anchor to sequence the whole virus. And once they had that sequence, then that allowed the screening of blood products. So a lot of hepatitis C infections before 1989 involved transfusion or blood product delivery from sources that were infected. So once they were able to screen that out of the blood supply, that did a lot to prevent further transmission of hepatitis C, although still new infections occur largely due to IV drug use. And a large number of humans, maybe about 3% worldwide, do still have chronic infection. But, just a quick, the genome is similar to what I described earlier. The 5' end is an iris rather than a cap. The protein organization is a bit similar. There's 2 envelope proteins instead of the membrane and envelope protein, and the NS5 functions are divided amongst these 2 pieces, NS5A and B, but generally the architecture and replication strategy is the same. So the pharmaceutical industry rode to the rescue. So the treatments for HCV had previously been adding exogenous interferon to patients, or a drug called ribavirin, which targets the RNA dependent RNA polymerase, but these were not particularly effective. The side effects were fairly nasty. A few years ago now, Gilead Sciences came out with a drug called Solvati, which is an inhibitor of NS5B, the polymerase. So yeah, that was approved in December 2013. And there are a couple others that were made by other pharmaceutical companies, and in fact Gilead has another drug which inhibits NS5A. I think we're not sure exactly how it works, but resistant mutants can be found in the NS5A gene, so we know it targets NS5A. And that's now the state-of-the-art HCV therapy is a combination of the NS5A and NS5B inhibitors. And they work really well, and they are incredibly expensive. This actually is a slide I got from Ken, just to emphasize some of the, the costs involved here. You could have a whole discussion about, is this warranted? The company spent a lot of money developing the drug, but is it actually available to, all the people who need it? A lot of people can't afford this. There are various programs, some of them by Gilead themselves, to, provide or defray the costs for individuals for whom this is a financial burden, but just to throw out there that, this is how the pharmaceutical industry works. Okay, so we're going to go back about 225 years or something like that now, and kind of get to the history of some of these things, and I have about 20 minutes, 25 minutes, so we'll see how we get through this. Okay, that's the molecular biology of the virus that we know now. But in Philadelphia in 1793, they knew none of this. They only knew of this disease, which would occur somewhat seasonally in the summertime. That involved, as described in this, text here, involved high fever, something they defined as the black vomit, which was actually coagulated blood that was thrown up. General jaundice, which is where the name yellow fever comes from. And that it was very often fatal. This disease remained a problem even in the United States for quite some time as sanitation became better. It began to subside a bit, but especially in the South and port cities like New Orleans, yellow fever remained a problem for some time. A lot of this involved importation of mosquitoes. The slave trade was a major cause of transmission. And as I said, the symptoms were quite often fatal. And in the late 1800s, a Cuban scientist named Carlos Finley actually first proposed that maybe this is due to mosquitoes. And this idea bounced around for a while. It had its advocates and detractors. But finally, this was sort of proved fairly conclusively due to the work of Walter Reed and his colleague Jesse Lazier and others who then in the very early years of the 20th century were able to identify that this was a viral agent. So at this point, the ideas of viruses were beginning to be understood as something that could be run through a filter that would normally take out bacteria, but then you would still have the infectious agent going through the filter, so those were viruses, and that the transmission needed to go from the mosquito to the human. Lazier, I think, showed this rather definitively by having one of his, just trying to maintain his experimental mosquitoes, fed one off his own arm. And then he got yellow fever and died. Yeah. And further controlled experiments demonstrated that if you maintained a mosquito-free environment, that even things like having volunteers, mostly I think members of the U.S. Army, sleep in the, the unchanged bed of somebody who had just suffered from yellow fever. They still wouldn't get infected if there was no mosquito vector to transmit that. So further work, not all of it involving human subjects, but some, have shown that the thing works this way. So you have a human who has yellow fever. There's a period of viremia where the virus is circulating in the blood. During that period, which overlaps but is not entirely defined by the illness, the mosquito, if it were to bite the victim, will acquire the virus. The mosquito is not just a vector. The virus infects and replicates in the mosquito, but generally this is not fatal to the mosquito. And then, so there's a period where that mosquito is not infectious, and then after a certain amount of time, then the mosquito refeeding can transmit the virus, and the cycle continues. So a major breakthrough involved the work of Taylor and Smith, who, this is in 1934 or thereabouts, developed a yellow fever vaccine. This was done by taking yellow fever and passing it through chicken cells. So they did this continuously for about 3 years. They would just infect the chicken cells, collect the virus that came out of it, and infect a set of naive chicken cells. And that, doing it over time, generally will result in mutations occurring in the virus. And you're no longer the virus is adapting to grow in chicken cells, which probably or often means that it's going to be less adapted to growing in humans. And this is what's found, originally tested in primates, that the virus was indeed attenuated, but it produced an immune response. Again, so this is requires the adaptive immune response, largely the antibodies produced by the B cells, and that so you could give this to people. They generally were not sick, mostly. And that so you have an effective vaccine that produces fairly long-term immunity. It's known that there's like 67 nucleotide and only 32 amino acid differences from the original strain. The exact mechanism of attenuation is still not entirely clear. It's still a live virus, and it's not recommended for certain immune suppressed individuals or the very young. So it's maybe not perfect, and that's why we don't generally use it in areas that are not endemic to yellow fever. But it's still an effective vaccine for controlling virus in third world areas. This is actually currently a problem because there is under-vaccination throughout a lot of the yellow fever endemic areas of South America and Africa. This is sort of showing the presence of yellow fever in 2010. I was recently at a meeting discussing, it's largely about dengue virus, but also includes other viruses like yellow fever. And some of the individuals from Brazil had discussed a recent outbreak that had occurred there just in the past year or two. So besides the lack of good vaccination in the whole country, the other problem involves one of vector control. So the major vectors for yellow fever and many of these other things is Aedes aegypti. which is highly domesticated. It lives preferentially with and in the houses of humans. It just breeds in domestic water containers, or tiny little bits of standing water are enough for the mosquito to lay its eggs. The secondary vector, Aedes albopictus, can also transmit these things. This is something of a broader host range, so it doesn't mind feeding off other mammals besides humans or birds. It's defined as peridomestic, sort of living in and around human areas, so sort of from the cities into the more rural areas surrounding them. And both these mosquitoes are capable of replicating and transmitting many of the flaviviruses, certainly yellow fever, dengue virus, Zika virus. So a lot of ways of controlling the outbreak of these things involves mosquito control, something that was accomplished around the 1970s to some extent. But then a lot of those programs have fallen by the wayside, and the mosquitoes have rapidly returned to many of the areas where they had been eradicated. Right, about 10 minutes, you think? Yeah, all right. Oh, we'll do dengue. So I've got dengue and Zika virus, but let's go with the dengue viruses, because these are sort of interesting. So the dengue viruses are actually 4 different viruses, dengue 1, 2, 3, 4, that are defined immunologically as related but different. So all the serotypes are defined by the disease they cause. That's dengue fever, which is a severe and sometimes debilitating fever that in some cases can morph into a more severe form called either dengue hemorrhagic fever that involves loss of vascular integrity and hemorrhage, and then in the most severe cases, dengue shock syndrome, which is potentially lethal and both hemorrhagic and shock syndrome generally require hospitalization for the patient to recover. So within the 4 serotypes, there's genetic variation that we call strains, and some of these strains seem to be more or less virulent than others. But this is, we can observe like sort of hotter or less so strains, but the actual mechanisms of how that works is still something of a mystery. Next. So these are, again, found throughout the tropical and subtropical world, within the Western Hemisphere, Central and South America, and they are kind of creeping up to the southern United States, and you'll occasionally find dengue virus sort of being transmitted locally within Florida and Texas, also found in Africa and Southeast Asia. Estimated close to 400 million infections per year, of which about 100 million of those are symptomatic and maybe 500,000 severe, requiring hospitalization. Just a thing from the CDC showing cases of Key West. This was in 2009, where you had a case, incidence of locally transmitted dengue, so not patients who were coming back from South America, but the mosquitoes in the local area were actually transmitting this virus. So generally it's believed that infection with one of these serotypes, once you get over that, you should be immune to infection with that serotype for a while, if not the rest of your life. And a lot of this data comes from experiments done on humans, again, volunteers, done by Albert Sabin, the oral polio vaccine guy, just at the end of World War II, when dengue became of interest because of the U.S. Army fighting in the South Pacific. And he observed that initially there was a sort of initially after dengue infection, you were probably immune to most serotypes, but this eventually waned to become this serotype-specific immunity, and this is how we began to define the different serotypes of dengue as to what, how one responds to different isolates of dengue, and they could be then classified as 1, 2, 3, and 4. An interesting observation is also that a secondary infection with a different serotype, say, for example, you had an infection of dengue 1 and then you got dengue 2 at a later time, that dengue 2 infection is probably worse than somebody who is just seeing the dengue 2 for the first time without having seen any other serotype at all. And the general idea of why this happens is something called antibody-dependent enhancement. Let's skip that. So antibody-dependent enhancement is the idea that when you are infected with one type of dengue virus, you mount an immune response to it. You now have antibodies that recognize that dengue virus. If you're infected with a different serotype, it'll see that other virus. The antibodies will bind to that, but not very well and not in a way that prevents them from infecting cells. So you have antibodies sort of, let's just say, loosely hanging on to these, the other serotype viruses. Then cells that recognize antibodies, macrophages, for example, within the body, that have receptors called Fc receptors that normally recognize antibodies and internalize what they're bound to. That's part of the immune response. Will take up these dengue viruses, but they're still active, and then they infect the macrophage, or there are other cell types involved in this too. So having a suboptimal antibody response, or one that was sort of directed against one serotype but is not very good against the other, makes the second heterologous serotype infection worse. There's a fair amount of evidence for this, and it's also been modeled in a mouse model of dengue infection, shown here. So this is a mouse infected with dengue 2. Shown in the open squares are just what happens. So this is sort of optimized for mouse passage. But if you add serum from dengue 2 to the virus, to these mice before viral infection, at least you offer some protection. More of them survive. This is survival curve. So this is percent of the number of mice that are still alive after the dengue infection. If you give them dengue 1 or 3 or 4 immune serum, shown here, they die much more rapidly than even in the case of no immune serum whatsoever. And that's largely due, shown here is neutralization of them. So shown, if you just look at this bar here, which is neutralization of dengue 2 by dengue 2 specific serum, this line is shifted to the left, which indicates less efficacy for all these other serotypes. So this has some implications for the idea of vaccinating against dengue virus. So there are a couple dengue virus ideas out there. This goes back to some of the molecular biology that I talked about earlier. So the most advanced, and the one that's actually being used in several countries, is this one on the top, which is called, originally CYTDV by Sanofi Pasteur, or now called Dengvaxia. And this is actually the yellow fever vaccine. So they knew the yellow fever vaccine virus was a reasonably good vaccine. So they took the sequence of this virus and put the PRM and E proteins into it. And in culture, these things can grow, so you can make more of them. And then they're fairly attenuated, so they're not dangerous to patients when you give it to them. And then they'll make antibodies to these proteins, which is where you want your antibody response to the viral envelope, okay? So this idea has been around for a little while, and they tested it and worked in mice and non-human primates, and eventually went to very large clinical trials in dengue endemic areas, so in Latin America and Southeast Asia. So in Asia, they looked at children 2 to 14 years of age, enrolling a total of 14,000 kids in their study. And in Latin America, slightly older, and that was 21,000 individuals. And then you just follow these individuals and see who gets dengue and define, vaccine efficacy by sort of the hopeful reduction in the number of dengue cases in your vaccinated cohort, right? So it was okay, 50 to 60% efficacy. Maybe not as good as you would hope, but it did definitely worked. It's actually, this number is general for all serotypes of dengue. It's actually better for most of them, and dengue 2, for some reason, it didn't work particularly well, so that sort of brings the number down 50 to 60%. And if you can, reduce dengue infection in a certain number of individuals, then you might also argue that, you're helping to break the cycle of mosquito to human transmission, and the overall effect would be, even more beneficial than this number would suggest. So it's currently licensed in 13 countries. For use specifically in 9-year-old or older. This caveat came from some studies in the Asian cohort, which noted a slight increase in hospitalization among vaccinated children 2 to 5 years. So what was all that about? Why did it make things slightly worse? So that's dengue involved hospitalization. So the youngest part of the cohort who was vaccinated when they were infected with dengue tended to have worse outcomes than the placebo group. Oh, that's not good. But and then further analysis suggests that there is actually an increased risk of severe disease, not just in children, but any individual who is seronegative at the time of vaccination. So it's just that these are dengue endemic areas, and people growing up in them tend to have dengue at some point in their lives. So the older you get, the more likely you are to have experienced it at some point. So the younger ones were more likely to be seronegative, but if you look across a larger spectrum, now like all the way through adults with vaccinated individuals, it seems that anyone who has not had dengue before is potentially at risk for a more severe dengue infection, and this is possibly due to the ADE phenomenon that I described earlier, even though this is, does have representatives of all 4 serotypes. It's not clear if one of them is not working well, or if it's merely that you need something more than just the antibody response, like a T-cell response, which this particular thing is not going to produce, to protect against dengue virus. So I believe currently that the virus has been withdrawn from use in the Philippines. It still does give protection in a large number of, in the dengue positive individuals, and so research is ongoing, but it's sort of a bummer for the Sanofi Pasteur guys. My last virus is Zika, but I think I'm out of time. Is that right? Yeah, okay. Zika is interesting too, but next time. Yeah.
Thank you.